when we look at those vaccines, however, and when those antibodies are induced, it is mandatory that also CD4 T cell responses are induced. So if you want to have high affinity antibody responses against the protein of the pathogen, whatever you're vaccinating against, you need to have a robust CD4 T cell response, not only to induce affinity maturation, but also to be able to induce class switching and long-lived memory responses. Interestingly, therefore, 26 out of 26 vaccines, so virtually all licensed vaccines, induce CD4 T cell responses. And 24 probably work through the mechanism to induce high-affinity, long-lived antibody responses. But there are 2 vaccines that probably only function based on CD4 T cell responses, and that's tuberculosis and varicella zoster. So the classical role of CD4 T cells in the immune system, or the first role that has been described for CD4 T cells, is to help B cells to generate those high-affinity antibody responses. So these T follicular helper cells, they have recently gotten this name, T follicular helper cells, are not only important for the B cell proliferation and memory development, but are actually critical to induce this somatic hypermutation and the antibody class switching. And in HIV infection, with a lack of CD4 T cell help and CD4 cells, we believe that this is a part of the huge dysregulation of the antibody responses, this huge array of B cell dysfunctions that has been described by Susan Moore, and the lack of immune globulin class switching. But we know about for 20 years by now that CD4 T cells are also critical to provide help for CD8 T cell responses. The killer cells Ken was talking about earlier, or Watkins is studying in the non-human primates. So CD4 cells, it's known for a long time that CD4 cells provide some sort of signals to the CD8 cells that modulates the effector function of the cells, but also maintains them throughout the course of a chronic viral infection. And we know that the absence of CD4 help results in a lag of memory development. So CD8 T cells will not respond to an antigen again if there's not a presence of CD4 help, but we don't know at which time point this help actually occurs. Another so in CD4 infection, where we have the lack of CD4 T cell help, we have this, we think that this exacerbation of CD8 T cell exhaustion and dysfunction is actually due to the lack of CD4 T cell signals. And we also know that in the chronic phase of the infection, of HIV infection, we have a lack of memory CD8 T cell development. This Th1 subset is also critical for the macrophage activation. So the whole phagolysosome pathway is triggered through CD4 T cells, and we also believe that one of the major reasons for the comorbidity between tuberculosis and HIV infection is in fact the lack of the CD4 cells to activate the macrophages. Now CD4 cells have a vast array of different functions I'm not going to go into. So the Th17 cells that are important to recruit neutrophils and macrophages towards the infected tissues, the T regulatory cells that are rather dampening the immune responses and are important in the autoimmune field, and the Th2 and Th9 cells that are critical for the control of parasitic infection. And all of those different cellular subsets of CD4 cells have been associated with one or the other comorbidity in HIV infection, like the microbial translocation and lack of recruitment towards the gastrointestinal tract that has been described by Jason Branchley and Wefer Francini, the Iris syndrome as a cause of a Th17 and T regulatory cell disbalance, and of course all the parasitic infections that we see as opportunistic infections in HIV infection. Lastly, it has been suggested that CD4 T cells have the ability to directly kill virally infected cells themselves. But by now, we don't know what this actually would mean in HIV infection. It has been shown that Epstein-Barr virus infection, or one part of Epstein-Barr virus infection, control of Epstein-Barr virus infection is mediated by cytolytic CD4 cells. And we also think that JC virus infection is in fact controlled by CD4 T cells. But so far, we do not know what the lack of CD4 T cells or cytolytic CD4 cells in HIV infection would mean for those infections in AIDS patients. So what you can see here is that, that really three major parts of the protection against an infection is mediated through CD4 cells. We have the memory development. You want memory to have lifelong responses against the pathogen. So we have the CD4 help for B cell responses. We have the CD4 help for CD8 responses. Otherwise, we wouldn't be able to clear viral infection. And we have a mechanism of direct killing and direct protection through CD4 cells. And it's actually interesting. Actually, 2 months ago, there was a paper coming out in Influenza in humans that showed that the pre-existing CD4 T cell responses and not CD8 T cell responses is protective and dampens the severity of influenza infection. So I guess from this standpoint, it might be critical to induce CD4 T cells for vaccine design. So why don't we study HIV-specific CD4 T cell responses, given the important role in the overall immune system and the orchestration of the immune system? Well, it basically goes back to this study by Danny Dueck, and he didn't do anything wrong, but what you can see here is that 12 patients listed, and the red dots here, these are the HIV-specific CD4 cells. And the squares are the general memory CD4 T cell population. And what you can see here, that the level of percentage of infected CD4 T cells is always higher in the infected, in the HIV-specific CD4 cells than the memory CD4 cells. So that led to the conclusion that HIV-specific CD4 cells are preferentially infected and depleted. However, when you take a close look to the axis, it shows that only 0.1 to 5% of the HIV-specific CD4 cells are infected. In other words, 95 to 99.9% of the HIV-specific CD4 cells are not infected and not depleted. And he actually states in the manuscript that the majority of the HIV-specific CD4 cells are not infected lifetime. So you can plot the same data actually in a different way, just looking at the total number of infected cells per ml, and you see that the majority, or that the bulk of the infected population is within the memory compartment. By the way, a compartment that also comprises HIV-specific CD4 cells. So the majority of the infected cells are lying within the memory department and not necessarily within the HIV-specific CD4 cells. So we therefore wondered, when we have these HIV-specific CD4 T cell responses, how do they contribute to the control of HIV replication? We therefore decided to just first look across the proteome and just screen for all the different CD4 T cell responses in a cross-sectional cohort of controllers and progressors, and just asked what is the level of CD4 T cell responses. And what you can see here is that there's not only a fair amount of HIV-specific CD4 responses, but that patients who make a fair amount of HIV-specific CD4 responses have, in average, a lower viral load than patients who make less CD4 responses. So this is a viral load, and this is a breadth, so the number of different CD4 responses. And you see a moderate association, inverse correlation between the viral load and the breadth of HIV-specific CD4 responses. Now that was actually for me really surprising because I'm coming from the CD8 T cell field, and about 10 years ago a similar study was done for CD8 T cells, also looking at the breadth of the HIV-specific CD8 T cell responses and viral load, and there was really no association between the number of CD8 T cell responses and viral load. But for CD4 responses, we see a modest association with lower viral load with more CD4 T cell responses. Now, our question was then next, what is the region that is actually targeted by HIV-specific CD4 responses? Similar what we know from CD8 T cell responses, there has been a lot of studies been done to really nail down the different epitopes. We wanted to know what is actually targeted by CD4 cells. And what you can see here was that there is quite a clustering of the responses within the P24 and the P17 region, so within GAG, similar to HIV. But we also noticed that there is a cluster here in the envelope region and in the NEF region. We therefore wondered whether patients that are able to control viral replication are also targeting different regions of the virus compared to patients that are not able to control viral replication. So here you basically see the whole proteome again, and in dark blue, these are the patients that are elite controllers, so controlling viral replication below 50 copies per ml. And in light blue, these are the viremic controllers with viral load below 2,000 but detectable viral load. So what you see here, there's already a difference in the targeting between viremic controller and elite controller. So the elite controller, in the majority, are targeting this GAG P24 region and barely have any GP120 responses. In contrast, the patients that have a rapid progression to disease, here shown in dark red, almost exclusively target the GP120 region, but have no responses here in P24. And it almost looked to us that there's a balance based on viremia. People who have a high viral load target GP120 but no GAG, and vice versa, people who are controlling infection target GAG but not envelope. And so we decided to plot those ratios. And what you can see here, frequency or magnitude, that this is actually quite a good indicator of control. Patients who target a lot of GAG but very little envelope are elite controllers. Patients that target a lot of envelope but not GAG are rapid progressors or high viremic progressors. And the same here for the magnitude. Now, we were actually surprised about seeing such a strong protein specificity. So we wanted to know whether there's an association with HLA class II alleles. HLA class II alleles are very different compared to HLA class I, so I make it a little bit simpler than Ken. I was actually hoping you were touching base on that, but so he was going in depth into this pathway, showing that CD8 T cells recognize their antigen through the MHC class I pathway, and the HLA alleles are HLA A, B, and C. In contrast, CD4 T cells recognize their antigen through the MHC class II pathways, and this is HLA-DR, DQ, and DP. And the DR alleles are the most polymorphic, so they're most variation within this molecule. What Ken described as a hot dog with a bun, he was basically showing that it was really restricted how the peptide can lie in the MHC complex, MHC class I complex. Well, the MHC class II complex is a little bit different. It is rather open surface, and the peptide can lie on top of it. So this allows for a lot of variability, not only in the length, because it is an open end and it can have up to 30 amino acids that form secondary and tertiary structure, but it also allows much more for sequence variability, as the peptide can move around and basically can try to bind in a lower affinity to the MHC. All that is not possible in a pocket, as the interactions or the length of the pocket doesn't allow for any other different binding. So what we did is, from the first study, what I showed you with the different protein specificities, we generated over 168 HIV-specific CD4 T cell lines, and we from 38 patients, and generated fibroblasts that were transfected with one single HLA-DR allele, so to determine whether the CD4 T cell line is restricted by one or the other allele. It's a little, because there's such a sequence variability, we had to do this method because it was one peptide can be presented by multiple different DRs, just because of this high levels of promiscuity. So we had generated a panel of 32 different L cells together with Alessandro Sette and asked basically for the restriction of the HLA class II responses. And I mean, what has been described to other in other infections, we were still surprised about it, was this huge level of promiscuity. So one peptide, this single peptide, was presented by over 11 different HLA-DRs. So it was not like in what Watkins showed last week, that one response is restricted by one MHC allele. We have in CD4 T cells that one peptide can be restricted by up to or over 11 different HLA-DRs. So this level of promiscuity has been described for other pathogens, but we also see it in HIV infection. So there we were a little bit surprised, because on the one hand side, we see this huge level of association between gag responses in controller, envelope responses in progressor. But on the other hand, we see this big mess of different HLA class II restrictions that didn't seem to make very much sense that there might be any association between HLA-DR and control. But what we noticed was that there is actually a pattern in the HLA class II restriction. There were some HLA-DRs, like DR1302, 01, and 03, that were restricting quite a lot of different HIV peptides, while there were others, like 041 or 101, that didn't restrict for a lot of HIV-specific responses. So we had a few that were contributing a lot to the total CD4 T cell response, while others were barely involved in the HIV-specific CD4 responses. So we therefore wondered whether these responses are associated with control, while those are not. So our question was whether there's a difference, just a pure genetic difference of HLA class II alleles and HIV control. So we turned to a completely different cohort, and of over 1,000 patients where we had, that were treatment naive, chronically infected, where we had high resolution HLA typing. The analysis were adjusted for all the protective alleles that has been described before, like B57, B27, or rapid progression, HLA B35PX. And we asked the question whether the one or the other allele is more associated with protection or not. And it turned out that we had quite a fair number, but we didn't see any difference. I mean, it's important to point out, we didn't see any difference of one single allele compared to the other allele based on multiple comparison. But what we saw is that there was a trend from some alleles that were rather associated with protection and some alleles that were rather associated with rapid progression. And so we wondered whether this cluster here of the ones that are rather associated with protection or the ones that are rather associated with progression are actually involved in control. So we went back to our first cohort and stratified our HIV-restricted responses just based on those groups. And what we saw, that the ones that were associated with rapid progression were restricting about 10 different CD4 T cell responses. But in contrast, that were the HLA-DR alleles that were associated with progression restricted about 3 times as many CD4 T cell responses compared to the ones that were associated with progression. So we saw an independent, so both are independent cohorts, and we saw an independent effect of the function of the CD4 T cell responses with the protein specificity and with the different HLA-DR alleles. So to summarize this first part, the breadth of the HIV-specific CD4 responses is moderately associated with control, in contrast to the same study that has been done for CD8 T cells. We see a marked differences in protein specificity in controller and progressor, and we see differences in the number of peptides that are restricted by certain HLA-DR alleles. We also see that some HLA-DRB alleles that are associated with low viremia restrict a larger number of peptides compared to HLA-DR alleles that are associated with progression. But what we also noticed, that those responses in HIV controller are not only interferon gamma secretors, but showed the ability to degranulate. Now, what does it mean to degranulate? When a let's go to the CD8 cells. When a CD8 T cell is trying to kill in a virally infected cell, it brings out the effector molecule that are kept in a vesicle. And this vesicle on the cell surface, we can detect that has a different coating than the cellular membrane. And on this vesicle, we have LAMP1 and LAMP-B, what is also in the CD nomenclature as CD107A and CD107B, and we can detect those structures. And so we basically, with the detection of CD107A, we can detect the mechanism of degranulation, so the mechanism that something was brought out like perforin or granzyme or any of those effector molecules. And that's exactly what we see here, and those vesicles are loaded with perforin, granzyme A, and granzyme B, and not really granzyme K. So this is really surprising. We have CD4 helper cells that have the ability to degranulate and kill what has been associated with natural killer cells or CD8 T cells. So we wondered whether CD4 T cells are directly involved in controlling viral replication through killing mechanism. So we designed the following experiment. We chose subjects with very high viral load during acute infection, 3 million copies on average, that developed a high viral set point. All were treatment naive. Then we had a similar group of patients also with 3 million copies per ml on average that were not different at the 2 months time point, but then something happened, and they started to control viremia about a log lower. And here are the values, so you have the same average, almost the same average viremia, no difference at 2 months or baseline, but then we have a significant difference in viral load at both at the 6 months and 12 months time point. It's also important to point out that there's actually no difference in both of the groups in terms of CD4 T cell count. We first look at the interferon gamma secretion in both of those groups of the HIV-specific CD4 cells. And what you can see is they have very low level of CD4 T cell responses, but we see an initial decrease in the CD4 responses towards the 2 months time point, and then low levels of those responses. And in the patients that reached a high virus set point. In contrast, the patients that were able to control after the 2 months time point showed at the 2 months time point expansion, significant expansion of the HIV-specific interferon gamma secreting CD4 T cell responses. And it's important to note that this differences is detected before we see any differences in the viral loads. So we next, of course, you asked what the CD8 T cell responses are doing in those two groups. So just quickly, we never detected why we see a significant expansion from the first time point to the last time point, of course. And we see a temporal association of the emergence of HIV-specific CD8 T cell responses. We do not see any differences in terms of HIV-specific CD8 T cell responses at any time point. And it rather looked here at the 2 months time point that there's a higher level in the ones that reached a high virus set point, but it's not significant. So we next wondered whether these are cytolytic CD4 T cells, so have the ability to degranulate upon stimulation. Again, in the patients that reached a high virus set point, we see an initial decrease and very low level of the ability to degranulate. In contrast, the patients that were able to control viral replication, we see at the 2 months and 6 months time point a significant expansion of the cytolytic or degranulatory CD4 T cell responses. And again, this happens before we see any differences in terms of viral load. So now we wanted to know whether those cells are actually really able to kill virally infected cells. So we didn't want to activate the CD4 T cells because activation of the CD4 cells creates target cells for HIV. So we had to come up with a little bit of a different assays. So we used macrophages as targets. We first generated macrophages, infected it with a slightly different virus, used SIV VPX to get high levels of infection of macrophages, and then took CD4 T cells from infected patients directly in ex vivo and incubated it very shortly for 36 hours. But it's directly ex vivo. And then we, so you can see here that we get quite a high percentage of HIV infection of macrophages. 93% of the macrophages are infected. And when we add now the autologous CD4 T cells, we see about a 20% decrease in the factor to target ratio of 5:1. We see about a 20% decrease of the HIV-infected macrophages. And when we use a 10:1 ratio, we see about a 40% decrease of the infected macrophages. And we did that, of course, a couple of times. In just the target cells shown here, we get a very high levels of infection. And then we can see a dose-dependent effector to target ratio decrease in the level of infected macrophages. And we did the same experiment to show that it is not non-specific. We did the same experiment in HIV-negative patients, and we didn't see any decrease based on the CD4 T cell responses. Now we wanted to also show that this is an HIV class 2 dependent pathway. So we turned to CD4 T cell clones that we generated. So we infected now H9 cells as our target cells with HIV and used a normal viral inhibition assay. When we added our CD4 T cell clone, we see a 1,000-fold reduction of HIV viremia. Similar levels that has been described for CD8 T cells. But when we now block the TCR MHC class 2 pathway, we knew how the response was restricted. So we blocked this pathway with an antibody. We see an almost complete abrogation of the inhibitory effect through the cytolytic CD4 cells. So we now wanted to know, wanted to further characterize those HIV-specific CD4 cells. And we were asking what is contained in those granules. So we look at perforin expression, granzyme A, granzyme B, and granzyme K. There are a couple of more granzymes, but these are the major ones. And we asked how are those cytolytic CD4 T cells characterized? Interestingly, when we look in chronic infection, only at 2 months, 6 months, or 12 months post-acute infection, we see that about 20 to 30% of the HIV-specific CD4 cells have at least one or more of the effector molecules. But we don't see any difference in the patients that are able to reach a high or low, or reaching a high or low virus set point. In contrast, the baseline, when both levels of viremia were exactly the same between both groups, we not only see that the responses, that about 75% of the HIV-specific CD4 T cell responses express one or more effector molecule, but we also see that there's a significant difference between both groups, how they look like at baseline. And this is basically due to the orange arc, which is granzyme A. So what you can see here is that patients that reach a low virus set point, we see at baseline really high levels of granzyme A in the HIV-specific responses, in contrast to the ones that reach a high virus set point. And it looked to us that the level of granzyme A in HIV-specific CD4 T cell responses could almost predict disease outcome in this group of 11 patients. Well, now we thought, well, 11 patients is a pretty small number to make such a statement of prediction. So we turned to a cohort, completely different cohort of 26 acutely infected individuals. We were completely blinded, so we didn't know whether they were able to control or not control viral replication. And we measured at baseline, at peak viremia, the levels of granzyme A in HIV-specific CD4 responses, and wondered whether the level of granzyme A can predict disease outcome. And here we split them in granzyme A and granzyme A high and granzyme A low group. That's the time point where we measured the granzyme A levels in the HIV-specific CD4 responses. And you see that in average, about after 300 days, the granzyme A low group had the CD4 count drop below 350 cells per microliter. In contrast, the patients with high granzyme A levels in acute infection at baseline developed about a year later, dropped about an average a year later below the level of 350, and that was statistically significant. Similar when we used a different readout. I mean, we unfortunately we couldn't use. We had to go with what the clinician decided to do with the patients. But when the patients had to go on hard treatment, antiretroviral therapy, it was also about a year later, or when the viral load went above 100,000 copies, we again see a significant difference between both groups. So why is this for vaccine important? I mentioned earlier the influenza paper, but we have also a little bit of evidence from the HIV field. You probably all have discussed in depth the RV144 trial that showed a moderate protection of 0.04 with modified protocol analysis. So the patient that received the ALVAC-HVAC vaccine were doing about 30% better than the patients that received the placebo. This ALVAC-HVAC vaccine induced non-neutralizing antibody responses, and that's a major focus on that. But what is only presented at the conferences so far is that it's also inducing quite high levels of CD4 responses, especially here against the envelope region. And the further characterization of those HIV-specific CD4 T cell responses showed that they not only have an Th1 phenotype, but have also quite high level of cytolytic activity. And which might be that this is also a mechanism how the RV144 vaccine might have acted or brought the low level of protection. So to summarize this part, I've shown you an early expansion of HIV-specific CD4 T cell responses in HIV controller. I've shown you that cytolytic CD4 T cells are detectable in HIV infection, but enriched in controllers. We see that HIV-specific CD4 T cells with degranulatory activity expand early in acute infection in those that achieve lower virus set point. And we see that the enhanced levels of coenzyme A expression in HIV-specific interferon gamma secreting CD4 T cells are present at baseline in patients that are subsequently controlled. Now, what about CD4 help for antibody responses? One of the classical roles of CD4 cells, so and one of probably the most complicated pathways in terms of HIV-specific CD4 T cell responses. So let's go back and review a little bit how the pathway, or what we think, how the pathways of CD4 help for B cell responses, antibody development, is actually working. We have a naive cells that is attracted by the dendritic cell and is getting presented the antigen of the respective antigen. The CD4 T cell is being activated, and for T follicular helper cells, in particular IL-6 is an important pathway. Now this activation leads to the differentiation in T follicular helper cells. Now the T follicular helper cell is characterized through CXCR5 expression, PD-1, and the transcription factor is BCL6, B-cell lymphoma 6, the same that is associated with the cancer and B-cell lymphoma. Now, CXCR5 is also the chemokine receptor for CXCL13, and which is secreted by follicular dendritic cells, and therefore attracts the T follicular helper cells to the border, so we are in the lymph node, by the way, to the border of the follicle and the T cell zone. Now the T cell can't just enter a follicle. So you will never see, by the way, CD8 T cells in a follicle. The CD4 cell ends at the border to the follicle, and then that starts to interact with the B cell, the antigen, so the respective B cell that represents that particular antigen. So there's a multi-layer interaction between T follicular helper cells and B cells, and it's not very well understood so far. So we have on the one hand side the interaction between the TCR and the MHC class II, but we have also the important signals that come through CD40 ligand and CD40, the ICOS ligand, SLAM, and SLAM receptor. And all those receptors form a very tight and very prolonged interaction. So for quite a long time during this process, they don't let each other go. So actually the B cells confirms in the T follicular helper cell the expression of BCL6, while the BCL6 is inducing BLIMP1 in the B cell. The T follicular helper cell is inducing BLIMP1 in the B cell. Now, what the B cell is doing, it basically drags T follicular helper cells into the follicle, and then they start to form a germinal center. And that's a center reaction for every vaccine design, is this germinal center between the T follicular helper cells and the B cell. And the B cell starts to proliferate. It is induced antigen immune globulin class switching through the signals of different cytokines, and finally differentiates into memory B cells and long-lived plasma cells. However, one important thing is what we don't know yet is we don't know which signals leads to a plasma cell. We know IL-21 leads to a plasma cell differentiation, but we don't know what the T follicular helper cell is actually doing, and we don't know how we get memory. We know that without T follicular helper cells we don't get memory, but we don't know what signals are necessary to induce memory in the B cell, in the B cells. Now, this is basically how it looks like. We have here the T cell zone outside of the follicle with all the T cells. Then here a huge array of the follicle with all the different B cells. And here's the germinal center where the CD4 cell is with different B cells, and the B cells proliferate on that. Now, what happens to the CD4 T follicular helper cells in HIV infection? Are they depleted? Are they expanded? Are they dysfunctional? we don't actually know until now, because it was not really a part of intensive investigation. The problem also with studying CD4 T follicular helper cell B-cell interactions in humans is that it takes place in humans. It takes place in the lymph node. So what we had to do, we had 26 patients and 7 HIV-negative patients where we resected, surgically resected lymph nodes from the axilla of those patients. So they had to undergo surgery that we were able to receive a lymph node. And we assessed the HIV-specific T follicular helper cell responses in those patients. Now, they have not been defined thus far in human lymph nodes, so we had to see what they are actually expressing, and we found that also what has been shown in mice is that the CXCR5 positive PD-1 high population are also the ones that express the transcription factor BCL6. And you also see that those responses are not present in the periphery. So those cells are only found within the lymph node. And here again to confirm, this is a log scale that there might be very small levels of T follicular helper cells in the circulation. I don't know if they're there or not, but there's a high levels of those cells within the lymph node. However, in HIV infection, we were really surprised to see that. While in HIV negative, we have about like an average 1% of the CXCR5 positive PD-1 high positive cells, we see a significant expansion of T follicular helper cells in the lymph node. This is based on CXCR5 PD-1 high, but we also see that the transcription factor within this population of the CXCR5 PD-1 high is associated with the level of viremia, with chronic untreated having the highest, this is HIV positive non-treated, have the highest level of BCL6 expression. Now, we also saw that this was mainly driven by HIV-specific T follicular helper cells. So we saw a large expansion of HIV-specific IL-21 secreting T follicular helper cells in chronic untreated patients, also here for interferon gamma, that was significantly higher compared to the periphery. And again, this was the majority of the expansion that we saw within the lymph node. So we expected, so I really expected that they were gone. We saw the opposite. We saw that the HIV-specific T follicular helper cells are expanded. Now, why don't we develop then more broadly neutralizing antibodies? And that might go back to the role actually of T follicular helper cells. So our question was, how does this expansion of T follicular helper cells impact the B cell compartment? So these are B cells within the lymph node. It looks a little bit different in the periphery, but what you can see, we use the BM1 to BM7 classification of different B cell subsets, but what you can see here is in the HIV positive a large level of plasma cells. These are plasma cells that are barely present in HIV negative individuals. Same here for the germinal center B cells that are not barely present in HIV negative. And then we have this decrease in the memory compartment that is largely increased in HIV negative patients. Oh, I highlighted that. So here, to put it down to statistics, we see from HIV negative a large increase in germinal center B cells, the decrease of memory cells or late memory cells in the HIV positive patients, and we see a large increase of plasma cells in chronic HIV or HIV positive patients. And we also see, while we can't put it down to a mechanism, we can only look at correlations in humans here, we see that those is associated with the level of T follicular helper cells in the patients. So the germinal center B cell expansion is associated with an expansion of T follicular helper cells. Same more moderately for the plasma cells. And we see that the decrease in memory B cells is also associated with the expansion of T follicular helper cells. What we also see is that the expansion of T follicular helper cells is associated with hyperimmune gammaglobulinemia. So non-specific secretion of low affinity antibody responses. Now let's, just to be cautious, we can't put this down to a mechanism that this is really the pathway of what is happening, but it biologically it would make sense. So let's put it down to a model again. We have this big level of immune activation, and Alan is going to talk later about this, that through either directly HIV, and so the development of T follicular helper cells is not only driven by the TCR-MHC interaction, but also IL-6. We believe that this large outburst of a constant IL-6 leads to an expansion of the T follicular helper cell compartment. So T follicular helper cells before that were the limiting factor for the development of high affinity antibody responses. So they were able to be actually very selective which B cell they are going to interact with. But now they have, they basically have the ability to interact with every single B cells. So we have now an increase of non-specific and low affinity interaction between CD4 cells and B cells. And we also see this outburst of IL-21, and IL-21 we know leads to the development of short-lived plasma cells. And that's exactly what we think is happening through the IL-21, that we have a premature development of the B cell or the antigen-specific B cells towards low affinity plasma cell responses that leads to the high secretion of low affinity immune globulins. So to sum up the last part, I've shown you that HIV-specific T follicular helper cells are expanded in chronic infection and are not depleted. We see that the expansion of TFH cells is correlated with the skewing of B cell subsets in chronic infection, and that this is also associated with hypergammaglobulinemia in chronic HIV infection. So a lot of people were involved in all those studies. This is my lab. But a special thanks to Alessandro Sette, who helped us a lot with the generation of the different L cells, Mary, who helped us with all the HLA class 2 alleles, Bruce and Marcus and Alicia for their help with the cohorts, and of course our funding, which is all NIH. And thanks a lot to all of you for listening and having me here.